Dear students, greetings to all. In this module, we'll discuss about the concept of linkage proposed by Thomas Hunt Morgan when he had carried out the experimentation on Drosophila melanogaster. Now, the genes which controlled the trait, that is the brown eye, the brown body, red eye, the yellow body, white eye, and the wings, whether that is miniature or the normal, they are present on the X chromosome as can be seen over here. There are no genes for the body color or the eye color or the wings present on the Y chromosome. So they are X-linked trait which Morgan had studied. Before carrying out the experimentation, Morgan was not knowing about the linkage or whether these genes which control the body color and the eye color, whether they are present very close to each other. But then after carrying out the experimentation, when the result differed from the Mendelian result, he came to the conclusion that it is because the genes, they lie very close to each other. That means the genes are linked, and therefore the probability and the possibility of recombination will be less because the distance between the genes is very less, and therefore they will not undergo recombination or will undergo very minuscule crossing over. The chances of crossing over are minimal. So let us try to understand the cross which was carried out by Morgan. So the trait which he had selected, that was body and eye color. Now brown body, that's the dominant trait, and red eye color, that is again the dominant trait, which is present in the male. It is denoted, the brown body is denoted by a Y plus and the red eye by a W plus. So the plus over here indicates that it is a wild type, means it is the dominant trait. It refers that it will get expressed in every generation. The recessive trait for body is yellow, and for eye color it is white, and it is found in the female. And we can see that the genes which are encoding these traits, they lie close to each other. They lie very close by on a particular loci on the chromosome. And the distance between them is very less, and therefore they are referred to as the linked genes. Before we produce the F1 generation, we'll have to go in for the production of the gametes. Now the gametes which will be produced from the male, they'll be X and Y, having a Y plus and a W plus. That is the gene which is encoding the body and the eye color on X chromosome. There is no gene which is encoding the body or the eye color present on the Y chromosome. So we'll keep it like that. From the female, we are going to have two X chromosome, but then in that case, because both of them are similar, therefore we'll take one X having the genes Y and W for body and eye color. Now we have got these gametes. We'll carry out the crossing between them so as to obtain the F1 generation. So first we'll cross this X with this X, and then we'll carry out the crossing of this X with this Y. So the result which will be obtained, that is when we cross this X with this X, we'll obtain a Y plus Y, W plus W. So because we are having both the dominant gene, that is Y plus and W plus, therefore Y plus will get expressed, so that Y plus refers to the brown body and W plus the red eye. So we'll get the individuals having brown body and red eye. Here we are having the recessive gene for yellow body and white eye, and therefore this recessive trait will get expressed because there is no gene on the Y chromosome. So we have obtained the parental trait, that is brown body, red eye, and yellow body, white eye. But then we have obtained this trait in female, as we can see that there are two X chromosomes. So this dominant trait has got incorporated in the female now in the F1 generation, and the male has got the recessive trait, that is yellow body and white eye. So as to obtain F2 generation now, we'll carry out the production of the gametes. Now the gametes which will be produced from the F1, there will be 6 gametes which will be produced, 6 combinations. So then in that case, the combination 1 will be that of Y plus W plus, the other of YW, that's the recessive one, the other again of the YW from the male side, and this Y which is not having any gene for the body and eye color present on it. If in case, though we know that the genes are linked and there is a very less probability or possibility of crossing over, but then if in case crossing over takes place, it will take place between this Y plus and W plus, giving rise to this Y plus W, and this Y and W plus, giving rise to this Y and W plus. So this what we obtain is the recombinant and the two traits which we obtain, that is the parental trait. Now we'll carry out the crossing of the male with the female. Now the male, it is having an X chromosome, so we'll cross this X first with this X, then this X with this YW, again this X with this Y plus W, again this X with Y plus Y and W plus. So we'll obtain these probable combinations in the F2 generation. Then again the crossing of Y will be carried out with this X chromosome, this Y with this X. So similarly we carry out the crossing of this Y with this 1, 2, 3, 4 X probable combination, and so we'll obtain the combinations as depicted over here. So here we'll obtain that in F2 generation we have got the wild type and we have got the yellow-white, that means the yellow body and white-eyed individuals. So we have obtained the parental trait, that is brown body red eye female, and we have also obtained yellow body white-eyed individuals, both male as well as female. Here what we obtained, that is brown body and we've got white eye individuals. So brown body and white eye, that's not a parental trait. Again a brown body white eye male, that's not a parental trait. And then again we have got this red eye and yellow body female and red eye and yellow body male. So that's not a parental trait. So here on this side, that is on the right hand side, these individuals which we are obtaining, they are the recombinants which we have obtained. And this over here, these are the parental combination. So Morgan, he deciphered through this crossing that just 1.3% of the recombinants are produced and 97.8% of the parental combination is retained. Now this results a deviation from the result of Mendelism, the crossing which he had carried out, and thus it gave rise to the concept of linkage. It means that there are certain genes which are present close together, which are referred to as linked genes, and they get inherited or transferred in association. They do not get separated or segregated during the process of crossing over. Thus, linkage, the strength of linkage is inversely proportional to the distance between the genes and also inversely proportional to the recombination frequency or the crossing over. So in this cross, Mendel had carried out the crossing between by taking the trait, that is body and eye color. In the next cross, he had carried out the crossing between the traits. He took the miniature wing also into consideration. About that cross, we'll discuss later on. And when we talk about those cross, because the genes were not linked and therefore the recombinants which he had obtained, the percentage was much more, is because the genes were not linked. Here, because the genes are linked, the recombinants which are obtained, they are very less, just 1.3%. So in this module, we have discussed the concept of linkage in detail. Thank you.